Hi, everybody. I'd love to introduce Juan Karlusich from MIT, who's been working on, well, tracking the state of marine ecosystem using DNA and, like these big analytics methods. Welcome, Juan.
Thank you, Greg. Thank you for the invitation and the opportunity to show part of my work. Okay, so here I want to show you some pieces of work in which I have been working in the last few years that are mainly about improving our understanding regarding the diversity patterns and the adaptation patterns of marine plankton at global ocean scale, and using the integration of massive and diverse data sets. So let me go more deeply about this. So as a bit of context about the system of study, the plankton, they consist of the microbial population of the oceans that goes from viruses to bacteria to small eukaryotes, and they are very diverse in terms of morphology, size, and function. And regarding their importance, we can mention that the majority of the biomass in the ocean correspond to marine plankton. The rest correspond to animals like whales, fishes, and so on. Part of the plankton has the capacity of photosynthesis. They are called the phytoplankton, or also the marine microalgae. They do as much as photosynthesis as do plants on land. So they are key for the ecosystem of the whole planet. And this explains why they are, let's say, the basis of the marine food webs. They have impact on human activities because they modulate the fishing stocks. They influence activities like aquaculture and tourism. Part of the organic carbon that is generated at the surface of the ocean by plankton or by the phytoplankton and thus sinking towards the deep ocean, and they can remain there for centuries. And this process is called the biological carbon pump, and it has an impact on the climate of our planet in the long term. So these are the characteristics of general characteristics and features of plankton. As I mentioned, they are very diverse, including they are very diverse in terms of size. And this is a cool example that you can find on internet of an electronic micrograph that at first shows a small animal called copepods that are very abundant in the plankton. Then you can see over the copepod is like a cylinder that is a microalga called diatom, and over the diatom you see a bacteria. And in theory, we can go and zoom in a bit, and we can found maybe viruses over the bacteria. So you can see that plankton varies by orders of magnitude in terms of size, even though the whole plankton are, we cannot see the plankton by the naked eye, but they vary a lot. They have a huge diversity in terms of size. And this is important, as you will see, in terms of how to sample the plankton in the seawater. So most of my work consists of the mining of the datasets generated by the Tara Oceans expeditions. The main datasets were generated by the first routes of this expedition that were carried out between 2009 and 2013. And you can see that they collected seawater samples from the main ocean regions, including the Mediterranean Sea, Red Sea, Indian Ocean, South Atlantic part of the Southern Ocean, the Pacific, the North Atlantic, and finally the circumnavigation of the Arctic. An important characteristic of the protocols for collecting the plankton samples implied the use of a serial filtration system in order to collect planktonic samples enriched in different plankton size. As smaller organisms tend to be much more abundant than larger ones, for those larger size fractions, it was collected higher volumes of seawater. For each planktonic size fraction, they were generated different type of datasets. For example, it was the planktonic cells were broken, and then the released DNA and RNA were collected, isolated, and collected and sequenced. And in addition, they were using different technologies of imaging, including, for example, confocal microscopy. These technologies allows to detect the morphology of the plankton. Some of the technology consist of in situ imaging devices that were in the water and that collected images in real time, where others consist of for others the samples, the planktonic samples were fixed on board and then analyzed on land in facilities in Germany. And then different type of environmental physicochemical parameters were determined, including nutrients, temperature, and so on. So it's a very nice data set for understanding the ecology of plankton, doing modeling, and different type of activities. So my idea here is to show 3 examples that target different type of planktons, of planktonic functions, that are a good example of how the integration of these diverse datasets, including DNA and imaging, as well as other datasets, were used to inform about the biology of these organisms and the ecological implications. The first work is about a group of planktonic organisms that are called the nitrogen fixers. So, but before that, let me explain a bit about the problem of the nitrogen in the ocean. So most of the ocean is limited in nitrogen. Nitrogen is an important component of our proteins, of lipids, of DNA, of different type of cellular components. And when you take a seawater sample and you add some, you fertilize them with a nitrogen compound, in many cases you see a blooming of the planktonic populations. So, and that's indicate that nitrogen was a limited nutrient for that seawater sample. During this kind of experiment, it was detected that most of the ocean is nitrogen limited. This is also occurs on land and in agriculture. So the study of the organisms that are capable of fertilize the ecosystem is very important from the ecological point of view and also for the biotechnological point of view. So there are a few organisms that are able to do this a kind of biological fertilization. They do that by a process that is called nitrogen fixation and consists of let's say the generation of bioavailable nitrogen using atmospheric nitrogen. And this is a process that is catalyzed by a very complex protein that is called nitrogenase, and it's very costly. It's a very costly, metabolically costly process. That's why only a few bacteria are able to carry out this process. This type of bacteria are called the nitrogen fixers. They are much more well studied in land environment due to their importance in agriculture. So, at some moment we decided to carry out their study in the ocean, because there are many gaps in the knowledge. And one important thing or feature of these organisms is that they have a huge variation in terms of lifestyles. Some of them are free living, others live in colonies or aggregates, and there are also many cases that they live in symbiosis, interacting with other species. And the symbiosis can be the attachment to other type of plankton, to the outer part of other planktonic cell, or it can be like more integrated with the host, as it is in the case of the example that I am showing in the picture, in which the yellow filaments correspond to nitrogen fixers, a kind of nitrogen fixer living inside a phytoplankton called diatoms. So the main questions that that we decided for this project is to determine who are the main nitrogen fixers in the ocean, where are they found, which are their different lifestyles.
So, question.
Yeah, sure.
I mean, I was just thinking in terms of nitrogen fixation on land. Are these the main fertilizers of other organisms? If they're the other organisms are nitrogen limited?
So in the case, for example, of symbiosis, the nitrogen fixer exchange offers nitrogen to the host, and the host sometimes offers some other compounds like carbonic compounds or other type of benefits like protection from pathogens or grazers. There are other cases that they are free living, they are not interacting with the host. But in both cases, either symbiotic or free living, they are always a leak toward the environment, towards their surrounding. So and also when they are grazed, this nitrogen is start fluxing towards higher trophic levels in the ecosystem. And so yes, that's why they are so important. I don't know if your question was related. I am understanding very well your question, Greg.
Yeah, trying to understand their position in the food chain, and I guess
Okay, yeah.
in that, and maybe it seems like the other source of nitrogen is runoff from farms. So, like, how does imbalance of this system affect the ocean ecosystem?
Yeah, there is. So these organisms are, yes, at the very base of the trophic level. Yes, and there are certainly some in the coastal regions of the ocean. For example, there are imbalances that occur due to the input of nitrogen due to human sources. And in those cases, sometimes it is generated, yes, what it called there is a blooming of plankton that is so fast that deplete the water from oxygen and generate what is called oxygen minimum zones. So this is also related with the cycle of the nitrogen, but beyond the nitrogen fixers, as the term, yeah. But yes, the balance of nitrogen in the ecosystem is key. Yes, so this was the idea was to have a global picture about them, how they distribute under different environmental conditions and gradients at global scale level. So as I mentioned, the idea was to integrate different type of data sets. On one hand, we have the DNA sequences obtained from the seawater samples. And on the other hand, in parallel, from the same seawater sample, we have 2 million images from confocal microscopy. So the idea was to integrate them. But we start analyzing directly the DNA sequences, the metagenomes, because the bioinformatic tools for analyzing them are more advanced, and also because we have reference databases that allow us to compare the environmental sequence with the sequence from organisms that we know. For this, the first step was to compile 2 different databases of these marker genes, what is called taxonomic marker genes. One is NIFH, is one gene that codified to the subunit of the nitrogenase. So this NIFH gene is used as a taxonomic marker for nitrogen fixers. And the other is the REK gene, that is universal present in bacteria, so it's a proxy of, a proxy of bacteria. So the metagenomic reads were mapped against these 2 reference databases, and the ratio of the reads for NIFH to the reads for REK were used as a proxy of the relative abundance of nitrogen fixers among the bacterioplankton populations. So as I mentioned, we have metagenomes from different type of size fractions. So it was nice to compare across the different size how the nitrogen fixers are distributed. So you can see that there is an increase or an enrichment of nitrogen fixers when you go from the lowest to the smallest to the larger size fraction. And in parallel, there is also a taxonomic shift. In the smallest size fraction, they are mainly present proteobacteria, whereas in the larger size fraction, they're mainly formed by cyanobacteria that are photosynthetic. So this is the way of interpreting this is that in the smaller size fraction tend to be enriched in free living organisms, whereas for the larger size fraction we are capturing those nitrogen fixers that are symbiotic with symbionts of larger hosts, and also those nitrogen fixers that form aggregates or colonies. The differences between the different Cs in terms of size fractions also occurs at the level of the environmental variables. For example, small nitrogen fixers tend to be more widespread in terms of nitrogen concentrations and temperature levels, whereas larger ones tend to be constrained to regions of low nitrate temperate waters. And this is very clear when we plot the biogeography for the different size fractions. You can see that for the smaller nitrogen fixers, they are widespread in terms of latitudinal gradient, whereas for the larger ones, they are constrained to tropical and subtropical regions. And this is very important information about to take into account due to the process of tropicalization of the ocean to predict how nitrogen fixers community will change in the future.
Question. I've not heard that term before. What's tropicalization of the ocean?
Well, there is the temperature in the ocean is increasing. There are and this is also generating increasing and expanding. So the subtropical regions are expanding towards higher latitudes, and this is generating also changes in the plankton communities that are in many cases plankton communities that you see in these tropical and subtropical regions are expanding in areas, whereas those that are present at high latitudes are being constrained.
Got it.
And then for the different size fractions, I mean, this slide is like, let's say, a recapitulation of the results that I showed in the previous slide, but I want to show that these results correspond to the main, what is called the traditional 5 main size fractions, let's say. But there is another size fractions that correspond to the seawater that pass through a pore of 0.2 micras. So, and this seawater, this size fraction sample is usually analyzed for the presence of bacterial viruses. But for this project, we were also interested in checking if there is also sequences for NIFH, and we found a high amount of these NIFH sequences from nitrogen fixers that seems to be proteobacteria. And the most this was surprising already, but it was even more surprising to find them very abundant in the Arctic, where other nitrogen fixers are very low abundant. So with this, we are giving some genetic evidence to support the existence of what we call ultra small nitrogen fixers in the Arctic, and there are new other groups and other expedition targeting the Arctic that are including this question to understand much better this organism or to isolate them. So in the result that I showed for the previous slides were focused in the analysis of environmental DNA, but as I mentioned, we also have imaging data. It was very challenging at the moment to analyze this data because because I mean the protocol for analyzing them consists of generating a training data set of nitrogen fixers that we know how they look, and then generate a machine learning model for doing predictions over the whole data set. The issue is that we don't have let's say, a reference data set, reference databases as we do have with DNA sequences, because the reference databases depend a lot of the imaging technology, and also there are many other constraints. Finding manually a few example images among the 2 million images was not possible in principle, because these nitrogen fixers are low abundant in the ocean. Even though they are key species, they tend to be low abundance with some exceptions, but in general they are low abundant. So what we did is to come back to the metagenomic sample in order to help us to filter in the data sets in order to do the manual selection of a few example images for a training set. So when we check again the metagenomic sample, we selected the three samples from different ocean regions in which we found a high abundance of the NIFH gene. And then manually we check in those three samples for the presence of nitrogen. We went again to the corresponding parallel imaging data for those three samples. And we check manually for images of nitrogen fixtures that we know how they look. We were able to obtain a few dozens of images that were used for generating a random forest model, run it over the 2 million images of the whole Tara Ocean transect for making predictions of nitrogen fixtures, and then we corrected the prediction manually and used them as a new training set for improving the model, and so on. We iterate this many times until we stop finding new images. And this was successful. It was successful so to see that the data between DNA and imaging match and were very consistent. For example, both datasets were consistent to discover to describe the global biogeography of nitrogen fixtures and discover new hotspots of nitrogen fixtures in ocean regions that are under sample. This includes the Indian Ocean, also the Madagascar Channel, the South Atlantic near South America, as well as part of the South Pacific. With the images, we have the advantage that we can understand the different lifestyles. So we found, for example, filaments of trichodesmium. These are cells that are attached one next to the other, forming these large filaments. Then we also detected filaments of calotrychs that are free living, as well as filaments of calotrychs living attached to the outer part of a diatom that is called Chetoceros. Other examples are filaments of Reichelia living inside some diatoms, specifically the diatoms Rhizosolenia and Hemiaulus. And then also colonies of Crocosphaera and Crocosphaera living inside the diatom called Climacodium. And also we detected some images of a nitrogen fixer called Eucine living in symbiosis with a phytoplankton called aptophyte, or a type of phytoplankton that are aptophytes. This is a special case because this Eucine, this type of nitrogen fixer called Eucine, there was recent support to claim that actually they are so integrated to the host that are already an organelle. They have been evolved into an organelle, so they are part already of the host, as a single entity. So just
the integration of imagery, to what extent can that guide lead us to like more global monitoring of these ecosystems?
Well, this is, I mean, at the ecosystem level, the nitrogen fixers cover huge amount of lifestyle. So I would say this is, for now, is some kind of a special case. There are also a few more cases that can be similar to this, but then we have other cases that the integration is more loose, as the one that I showed here, that are just symbionts living inside, or also symbiotes living outside, and that sometimes also can be living and free living. So at that level, I think that yes, it's at the level of ecosystem is a nice feature. But I think the most important issue that brings up this result of the very strong integration is at the level of, let's say, biotechnological implications, because also it can give you more clues to generate in the future plants with these organelles, plants that are able to fix nitrogen fixers. This organelle is called, this UCNA species now is called the nitroplast because it is an organelle. So in the case of plants, there are many plants that are able to form symbiosis in their roots with nitrogen fixers, with some bacteria. So there are some people studying how to go beyond that and make plants that directly have the organelle in their roots. And also there are implications in terms of studying the evolution of organisms, because this is a nice example of a recent integration, a recent integration to generate a new organelle. There are other organelles that are more ancient. For example, the mitochondria or the chloroplast were former bacteria that were integrated by a host cell, and eventually they become organelles. So the implications, yes, at the level of biotechnology and at the level of evolutionary biology are huge. Yeah.
So it sounds like you can start doing low carbon fertilizer, where it's the bacteria that are producing they're producing the nitrogen, but the hope is that if you observe them living like in these symbiotic relationships, you can engineer that for crops. It sounds like.
Yeah. There are also a huge amount of research with the symbionts in itself of crops, like in leguminous plants. There are some proteobacteria that forms symbiosis in the root of plants, and they are also already. I'm not an expert in that topic, but there are already a lot of research and understanding regarding that. But we can go, maybe this opens the door to go beyond that, and yes, directly integrate the bacteria into the plants at the level of becoming an organ. Yeah.
So and now the images you're showing right now, they're the types that you were analyzing in the study, right?
Yeah, these are examples.
So satellite imagery, you can like, to what extent is that usable for identifying blooms of a particular species? Like sargassum is visible, but I'm not sure if a lot of these are.
Yeah, that's a good point. Satellite images are used sometimes to obtain, for example, to see blooming populations. And in the case of nitrogen fixers, the most common blooming populations are of Trichodesmium, this one, because they under certain circumstances they tend to this type of filaments are either detected as free living or attached to each other, forming big colonies. And the colonies sometimes are so big that they can see you can see them by the naked eye. This in those cases, the blooming of these Trichodesmium populations are seen by satellites, and this is a good way of detecting them. For other cases, is more, the blooming is less intense or less localized. But there are also attempts to integrate the data that we see from, for example, with DNA technologies or with this type of microscopy technology with the satellite data, in order to parametricize the satellite data to analyze that. And yes, actually, I can go. I have some slides later about this.
Oh yeah, that'd be awesome. Thank you.
Go more deeply. So yes, and I will just mention some big conclusions of this analysis. For example, we were able to determine the most abundant nitrogen fixers at the global scale. In terms of cell abundance, there were heterotrophic nitrogen fixers, whereas in the case at the level of biomass, they were dominated by this trichodesmium, this species. And also the integration of DNA microscopy was very useful for not only as a way of intervalidation and detect more robust results, but also as a way of intercalibration and to detect, for example, biases in the DNA-based analysis. For example, the cells sometimes tend to vary or to have different copies of their genomes. This is called polyploidy. We detect in some cases some species of nitrogen fixers that tend to have higher polyploidy levels than other. For example, trichodesmium has many more copies of their genomes than Reukelia. And these are biases that are usually not taken into account in studies that are based on genetic information, based on DNA. And finally, we also suggest or give support to the discovery of new symbiosis in the ocean. One case was that we detected some metagenomic reads that were very similar of symbiosis that were so far reported in freshwater environments. These DNA sequences were detected in a size fraction that was consistent with the size of the host that we know from freshwater studies, and we detected them in different multiple and distant ocean regions. So it was very satisfying to see that an independent research group find after our publication also was able to find they carried out a targeted analysis and targeted expedition in order to isolate planktonic species and culture them. I was able to find this new symbiosis that in which we first give genetic support to their existence in the ocean. And also we detected this kind of images in many samples that were very abundant in phage sequences. And to explain a bit, these images that were from a pennate diatom, what is called a pennate diatom, but it was very, let's say, very interesting to see that this image, this pennate diatom has these green rounds, these green granules that was always present, and then also another group that was this isolating organisms in the ocean was able to isolate them and culture them and understand them, and to see that they correspond to a new symbiosis that is very similar to the symbiosis that occurs between with plants. It's similar to the symbiont. So yes, to go more deeply about this part of the work, as I mentioned, we were able to determine which were the most abundant nitrogen fixers in the ocean. We saw differences in terms of latitudinal gradients between free living nitrogen fixers and nitrogen fixers living in colonies and symbiosis, and this is important information in the context of climate change. And also we detected new hotspot of nitrogen fixers in undersampled ocean regions that is now used for designing new expeditions in these regions. And also we gave support to the presence of new symbiosis in the ocean. And also we found genetic evidence for the presence of ultra small nitrogen fixers in the Arctic. This is very interesting. But all in all, we found that nitrogen fixtures are diverse in all size fraction and global distributed with patchy biogeography. And this was an example of how to integrate this data from DNA and from imaging at a global ocean scale. And in addition to nitrogen fixtures, in another integrative work was focused in phytoplankton. So phytoplankton are the component of plankton that are able to do photosynthesis. Half of the photosynthesis they produce half of the photosynthesis of the global planet, and as I mentioned previously, they are very important in the regulation of atmospheric carbon dioxide due to this process called biological carbon pump, in which some organic carbon is sink toward the deep ocean. It's important to know that the phytoplankton are very diverse. So to compare with plants are very diverse at the level of species, but eventually all the plants came from have a unique origin. They came from a green or an ancestor related to green algae, whereas in the case of phytoplankton, they are the photosynthesis. They cover different branches of the tree of life, in the sense that they cover bacteria, some bacteria that are called cyanobacteria, that include species like Synechococcus, Prochlorococcus, Trichodesmium, Rickelia, as well as a wide and diverse range of eukaryotes that has gained the chloroplast through different processes of endosymbiosis. Among them, we can mention diatoms, green algae, dinoflagellates, aptophytes, etc. So the study and this diverse, this feature of that phytoplankton are very diverse also has a limitation in the let's say in the genetic analysis or the genetic service towards phytoplankton. Let me explain a bit more here. So when we go and obtain a seawater sample and isolate the plankton and broke the planktonic cells to isolate the DNA and sequence the DNA, this is called a genetics survey, and it's used for analyzing phytoplankton. But in general, they have some limitation because in general, what is used as a taxonomic marker gene is ribosomal a gene coding for what is called ribosomal RNA. And in this type of the protocol includes the amplifying the selecting taxonomic gene by a process that is called PCR that introduces some biases. But this is maybe too technical. But then other biases are related, for example, with the number of copy of these taxonomic marker genes per cell. In some cells, like in some green algae, you can find only one copy of the gene, but in other type of phytoplankton, like dinoflagellates, you can find 5,000 copies. So the introduced biases in the quantification of the phytoplankton populations. And also, as I mentioned, there have been many events in which of gaining of photosynthesis capacity, scatter events across the eukaryotic tree of life, and also many events in which the photosynthesis capacity is lost. So there are many cases of related species in which one of the species is photosynthetic, so is a part of the phytoplankton, whereas other species is not photosynthetic. And they are difficult to distinguish with these RNA genes, because these are housekeeping genes that are present in all organisms. And also it's difficult to cover prokaryotes and eukaryotes because these ribosomal RNA genes tend to be a bit different.
Question.
But yeah.
I'm actually I'm not familiar with ribosomal RNA. So why this variant, and is it more specific detection or easier to capture?
There is a biological variance that is related with many things. It has been, there is an association, for example, with the copy number of this gene with the size of the cell. This is a bit related because we need more ribosomes. Ribosomes are RNA are implied in the protein synthesis. So for a larger cell, you need more of these molecular machines, for example. And there are also, yes, this is one of the main explanation to explain the variability. Still, the correlation with this size and copy number is not perfect, and there should be other processes regulating the evolution or the selection of duplicating genes. And then these genes are present everywhere, so it doesn't matter if the organism is photosynthetic or not photosynthetic. So you can, let's say, infer that the organism, the specific, this sequence, this ribosomal RNA sequence, came from a photosynthetic organism, according what we know with reference databases, but they are not perfect. And then, yes, there are some problems regarding technical problems regarding the way that we detect these genes, and that is the PCR bias. This is more related with, because there is we need to this type of protocols target a specific this specific gene by we need to design some conserved parts of the gene in order to do what is called a PCI, that amplifying the gene, and then we sequence the amplicons. Yes, maybe this is too technical, but it's more a methodological part that is affecting also the quantification of phytoplankton. So the proposal was related to this, was instead of let's say analyzing or amplifying the gene with this PCR protocol, the proposal was to mine directly the metagenomes. Metagenomes consist of sequencing the whole DNA that we obtained from a seawater sample. So it's not affected by the PCR bias. And so we extract the metagenomic reads to a gene of interest. And in this case, we carried out a search for a gene that is single copy, only present in photosynthetic species and universally present in photosynthetic species, both prokaryotes and eukaryotes. And these genes end up being PSBO. This is a gene that codified by for what they call the photosynthesis 2 complex of the photosynthetic electron transport chain. But yeah, so that is a part of, is an important step in photosynthesis. So once we select the gene, we carried out the validation using the data from Tara Oceans, the microscopy and the molecular data. So as an example, these are the correlation between the microscopy data in the vertical axis and the ribosomal RNA genes in the horizontal axis. The first row corresponds to a size fraction, and the second row for a larger size fraction. Each column corresponds to a type of a phytoplankton group. And you can see that in general that for dinoflagellates and diatoms, more or less the correlation are okay. Still the points are scattered, and there is an for example, over representation of dinoflagellates in the ribosomal RNA gene. But the main problems correspond to other groups, for example, aptophytes that are under-detected by far. And here are the same correlation, but with PSVO. So the correlation between PSVO and the microscopy data. As you can see here that the correlation with the groups different from dinoflagellates and diatoms improves. And also they are more closer to the 1-1 relationship. And also this also happens for dinoflagellates and diatoms. We are more close to a 1-1 relationship. And then after this validation, we generated a curated database for PSVO sequences, and we released the database, and this is something that is now used by different independent groups for analyzing their own metagenomes. And in this project, we also we wanted also to analyze, come back to the Tara Ocean data, analyze the phytoplankton population with these genes in order to see the ecology of the phytoplankton, the ecology of the whole phytoplankton community. And you can see, well, the data in the bar plots and in the maps are essentially the same. The bar plots are an average of the whole Tara Ocean transect. But what I want to show is that in the smaller size fraction, there is a high amount of cyanobacteria, Prochlorococcus, and Synechococcus that are the smallest photosynthetic cells that we know so far, and the most abundant on Earth, photosynthetic cells, the most abundant photosynthetic cells on Earth. And then in the larger size fraction, there are high abundance of, let's say, eukaryotic phytoplankton, and also cyanobacteria that we already know that they form symbiosis or filaments, and many of them are also nitrogen fixers, the ones that I presented previously, like Trichodesmium and Rickelia. But it was very unexpected to find still high abundance of Synechococcus and Prochlorococcus, now in blue, dark blue and light blue, in the larger size fractions. So I came back to the imaging data, and I found many of these cases in which we found these cyanobacteria living as symbionts of dinoflagellates or forming aggregates. To have an idea here, the red channel corresponds to the autofluorescence of chlorophyll. So it means they have chlorophyll, they produce photosynthesis, and the size is according to the size of Synechococcus and also to the extent Prochlorococcus. And then, so those results were essentially from PSVO, and we check a few examples for imaging. And then, so now I am showing the result exclusively analyzing the confocal microscopy. And you can see that, yes, we detect a huge amount of these cases. In the case of symbiosis, they seem to be widespread in tropical and subtropical regions. And in the case of colonies or aggregates, they tend to be also detected in many ocean basins, but especially very abundant in a few samples, like in one sample in the equatorial Pacific. But the main message here is that this type of cyanobacteria, they were so far considered to be part of the smallest part of phytoplankton, but and they were considering that these cases of symbiosis and colonies were rare cases, but we are showing that they are general cases that are detected in widespread and highly detected. So we need to consider them as let's say, present in a wide range of, or they covering a wide range of the plankton size spectrum, and this has implication in the biological carbon pump, because I am simplifying here, but larger organisms and larger particles tend to have a higher to sink in a higher proportion towards the deep ocean. So we need to take them into account in our calculation of how the atmosphere CO2 is regulated by the biology of the ocean. So finally, we found, as some conclusions, we found that a gene that with improved correlation against microscopy, much better correlation against microscopy than for ribosomal RNA gene. We generated a curated dataset that is already being used by the community, by different scientific groups around the world. We found a high abundance of the picocyanobacterial aggregates and symbiont in the ocean, and this PSBO gene is a promising marker gene for molecular-based evaluation of the entire phytoplankton communities and in a quantitative way. This gene was also used by a project that was implicated Royal Hourani, a researcher from France, that is specialized in satellite data, as well as Marina Levy, also specialized in satellite data, and the group of Chris Bowler, in which we use the PSBO marker for the global and for let's say quantifying the global phytoplankton community across the different Tara Ocean geographical sites, and integrate them with the different variables obtained from the satellite data in order to generate an estimation of the global phytoplankton community for the main phytoplankton groups in global maps that cover the whole ocean. I mean, with the advantage of satellite data is that we can cover the whole ocean and also have high temporal resolution. And this is a very promising approach. Still, the main limitation is that we still have few points represent for PSBO representing in the ocean, but the good thing is that there are new metagenons that are being generated from new Tara Ocean expeditions, for example, that better cover the Pacific, also new expedition from the recent expedition from the Atlantic, and in addition, the boat is constantly unplanned. There are many plans to cover better other undersampled areas of the ocean, and PSBO had the advantage in the accuracy of the detection of the different taxonomic groups of plankton.
How do you differentiate these things? Do you have spectral imagery to get at the? Well, I shouldn't say because these are underwater. They, are hard to capture. So, what do satellites see about them?
Yeah, no, that's a good point. So basically, we detect from the whole seawater sample. We obtain the DNA and we do a bulk sequences of the bulk DNA. So all the DNA is mixture there, but with this gene is we use the sequences from these genes from organisms that we know that have been isolated and have been described, and we know they are to which species they belong, and we can do a global phylogeny and we can see where the environmental PSBO sequence are grouped inside this phylogeny of reference sequences. And so far, the phylogeny of PSBO is very consistent from what we know from the evolution of phytoplankton. So it seems to be a good method for determine the taxonomic names of the environmental sequences. At least working at a broad taxonomic level, for example, as in these maps, we are mentioning diatoms, dinoflagellates, green algae. We are dividing them at global, big taxonomic groups. This has the potential also to go more deeply in the taxonomy and go, for example, to a species level. But for that also, we will have to improve, in some cases, the representation of reference PSBO sequences from branches of the tree that are maybe not well represented. But at the broad taxonomic level, with the data that we have today, it seems to be working very robust. Yeah.
So yeah, that's useful. And as you do more of these surveys, you'll have more organisms. And I'm assuming you're say like there was this organism in the ocean, or at least 30% of this organism. You had a satellite picture of that same area. You know what color it is. So you can train a model. But then, what's actually visible about these organisms? Imagine if it's a giant bloom, easy pattern. If it's normal conditions, it seems difficult. It would be mostly normal color, or I could be wrong about that.
Yes. Actually for the let me see if I have.
Or it could be just you're detecting the kind of water that's a good host for this kind of organism, and you're not actually detecting the organism, like a species distribution model kind of thing, not a direct sensing kind of thing.
Yeah. Actually, the satellite variables that were taken were, let's say, chlorophyll A, but then also sea surface temperature, power, fluorescence, attenuation, coefficient, reflectance for the different points at the same point in which the Tara oceans were sampled.
Okay, and these are 4 kilometers. These are pretty coarse analyses. Yeah. But hyperspectral, is that what we're seeing here? I see several different wavelengths, so I wasn't sure what.
Yeah, I don't remember right now. It was mainly carried out by Roy El Jurani.
Okay.
But yes, there was different.
But it sounds like you're doing less of a direct detection and more like a map of ecosystems and the kind of ecosystems where different organisms thrive. Is this family of maps.
Yeah, that's a good point. Yeah.
Yeah, it makes a lot of sense. Thank you.
Yeah, thank you very much, Greg. Yes, I don't know how much time we have, or maybe
I think it'd be at the end of our slot.
Yeah, perfect. So this then at some moment I also was planning to tell about a very recent project a project that we finished very recently that was more focused on a type of phytoplankton that are the diatoms that I mentioned in some cases before, but I will skip it. But this basically is an example of also data integration between, in this case, molecular data, but also microscopy data. And I want to say, yes. So part of my, the research that I present here, that I present mostly here, was mainly from my previous postdoc in the lab of Chris Bowler in Paris, L'École Normale Supérieure. And now, in this diatom project, I didn't have much time, but I did it in some kind in collaboration with the lab of Chris and with my current lab in the lab of Sergei Ovkinokov in MIT. And well, this is part of my work more related with ecology, and also I am also doing some research more related with evolution and in the lab of Sergei. So yeah, thank you very much for your attention.
Great. Well, thank you very much. Have a great rest of your day.